Hello everybody, it's my pleasure to speak here today, and I would like to start with thanking the organizers of the meeting and in particular Allah for the invitation. I will be speaking today on mutational landscape of cancer and algorithmic methods developed by my group to study this landscape. I will start my presentation with a brief introduction to mutational processes in cancer, their signatures, and a brief discussion of what we hope to learn from studying them. Then I will discuss some algorithmic challenges and propose solution, focusing on the methods developed in my group. Finally, I also hope to convey some of the interesting lessons that we learned from those studies. Over cell lifetime, DNA of every cell continues to accumulate mutations. Those mutations are caused by endogenous processes, that is, processes within the cell that are stochastic, are also imperfect, as well as exogenous factors such as environment. Even in the optimal situations, it's been estimated that each cell can accumulate as many as 10 to the 5th lesions per day. Fortunately, the vast majority of those lesions are being very efficiently repaired by complex DNA repair mechanisms that cells involved. Only a very small fraction of those lesions remain unrepaired and then leads to mutations. Yet most of those mutations, again, are harmless so-called passenger mutations, and only occasionally the so-called cancer-driving mutations, that is, mutations that provide growth advantage to the cells that harbor them, emerged. However, the situation is often less than ideal, and there are certain environmental processes that significantly increase the mutational burden, such as smoking or UV light. In addition, there may be deficiency in DNA repair mechanisms can further aggravate the problem. Such deficiency can be acquired during the lifetime of the organisms and can be also genetic, such as often mutation in BRCA1. So overall, this is the interplay between the mutagenic processes, environmental processes, and this what we call primary mutagenic processes, and the DNA repair mechanisms that I refer to as secondary mutagenic processes that generally contributed to the mutational landscape of individual genome. So many factors can contribute to mutational landscape of an individual cancer patient. And the question is, given a sequence, a genome, can we go back and understand and decipher which mutagenic factors contributed to that particular, to the mutation in this particular genome? Why this is important? Well, first of all, understanding which primary processes may have contributed to the mutational landscape of an individual is important for prevention and tracing such environmental factor and understanding which environmental factor may be influencing a particular group of patients. Secondary processes, that is the processes related to DNA repair, understanding those processes is important as it provides opportunity for drug development. For example, it's well known that patients that have homologous recombination deficiency respond well to PRP inhibitors. It's also interesting to study the reaction of an organism to specific mutagenic exposure, and that we can do when we understand the mutagenic exposure of an individual and also say study the gene expression of this individual. Interestingly, such reverse engineering is to a large extent possible, and what makes it possible is the fact that mutational processes do not act uniformly on the genome, but rather different mutagenic processes have different characteristics. For example, it has been observed that for UV light, tends to produce a lot of mutations from C to T. So here in this experiment, you have comparative analysis of mutations in mouse cells exposed to UV light and not exposed to UV light. Not only you have much more mutation for associated with UV light exposure, but also you can see that those mutations are not the same, do not have the same distribution as the mutation before the exposure. So to provide additional detail and hopefully obtain a unique description and unique distribution for each mutational process, Alexandrov et al. Suggested to consider each mutation in the context of its flanking residue, and then characterize individual processes by the distribution of such triples. Indeed, for the UV light, not only C to T is a favorite mutation, but also this mutation preferentially occur when the C is being preceded by T. So now, of course, UV light mutations are well studied, and in general, we even don't know all the possible mutational processes. However, we have now defined what is signature, mutational signature, and mutational signature is now being defined for the purpose of this presentation as a distribution of mutation within such all possible triples, and hopefully each mutagenic processes will have different distribution. So even if we don't know all the possible mutagenic processes, once we formalize the concept of mutational signatures, we can ask the question that given a large set of patients and the mutational data, identify the optimal set of signatures that can explain the patient data. Under the assumption that the mutations in each patient can be explained as a linear combination of mutagenic processes, of the mutation from each mutagenic process that act on a cancer on a given patient genome, a natural mathematical formalization that allows us to identify such optimal set of signatures would be using non-negative matrix factorization. And this is exactly what Alexandrov and all did. Despite unsupervised nature of the non-negative matrix factorization, the signature obtained using this technique were very satisfying. Many of them could be directly linked to known signatures, to known mutagenic processes, and were in very good correspondence of what was expected of those processes. Signature 4, tobacco smoking, signature 7, UV radiation, signature 1 also correspond to well-known process, and many others. Other processes had an indirect suggestion what the signature may be corresponding to. Some of them were perhaps something that I will come back to as signatures, as a number of different signatures associated with one mismatch repair process, and we'll discuss that later. So although non-negative matrixization is not necessarily the only way one could approach this problem, because of this, satisfying results, those signatures obtained in this way have been widely accepted by biological community, and are typically used as the reference point in discussing of mutagenic processes in cancer. Okay, before I go to the algorithmic challenges, let me briefly summarize the key point of this introduction. So firstly, mutations are results of specific mutagenic processes. We are going to assume that different mutagenic processes are characterized by different mutation patterns, so-called mutational signature. For this talk, mutational signatures are defined as frequency vectors of mutation of the central nucleotides in each of the possible 96 triplets. Studies of mutational signatures lead to many interesting computational challenges. Today I'm going to discuss 4 such challenges. Those 4 are examples of some questions related to mutational signatures that my group was studying. The first of those questions may appear very simple. It is a question of identifying signatures that are present in a given cancer genome. So assume that we sequence a cancer genome, we know this is the definition of the signature, and would like to know for this particular genome which signature are present and what is the strength of each signature that is present in the genome. When we first look at this problem, we were surprised to note that for the same genome, different methods here shown in different color can sometimes provide quite different solutions. Interestingly, for our own decomposition method, signature estimation, which is based on quadratic programming and thus should be providing optimal solution, we also noticed that a small perturbation in the input data can sometimes lead to quite significant perturbation in the resulting decomposition. So it is the composition which is often unstable, and therefore some statistical framework of assessing probability p-value for each existence of signature is really needed. Fortunately, we could see that a simple bootstrapping method can go a long way. So using a very basic bootstrap-based approach, we have been able to provide p-value for the event that a given signature is present in a given genome with exposure level above specific threshold. And this table here shows the result for the breast cancer data. On the x-axis, you have the signatures that have been considered. In gray are the signatures that are shown by previous study to be contained in the genome of breast cancer patients, and each row here shows how many patients have contained given signature with the exposure above this threshold and p-value of 0.001. So based on this analysis, we can very confidently add 3 more signatures to the breast cancer patients, and in fact, signature 9 was later confirmed. The next question that we ask is this: given a mutation within a patient genome, are we able to decide which mutagenic process is responsible for generating these mutations, this particular mutation? To be more specific, assume that we have already performed the analysis described in before. That is, we know that a given patient genome of a given patient contains with high confidence signature 1 and signature 2. And we ask the question, looking at particular mutation from C to T, which of those 2 signatures generated that mutation? And that is not an easy question. Since signature 1 tends to generate a C to T mutation, we can speculate that signature 1 is more likely, but that's actually not always the case. So how we can approach this question? So in order to have a better understanding of which mutational signature could generate a particular mutation, we have to turn a little bit to the biology. And the relevant observation here is that many mutagenic processes are known to act in the context-dependent manner. For example, ApoBec enzyme is known to mutate a single-stranded DNA. So if DNA is in the single-stranded conformation being exposed, it's likely to be a target of ApoBec enzyme. So if we have larger regions exposed of single-stranded DNA exposed, then we would expect that there will be higher density of ApoBec mutations in that regions. Similarly, a process known as transcription-coupled repair is preferentially repairing damage in the genome, in the transcribed portion of the gene. So, of course, again, you will have a local dependency of which mutation can and cannot happen within that regions. So we hypothesize that neighboring mutations are likely to be a result of the same or related mutagenic processes. And based on that, we propose a hidden marker-based model to assign mutations to specific signatures. The method, called Sigma, has been developed in collaboration with Roded Sharan and Mike Leiseson's group, and briefly works as follows. So the input to the method is the set of signature present in a given genome, say identified using the methods that I previously described, and the set of mutations, indexed set of mutation within the given genome of the given patient. We also partition those mutations into two groups. The so-called SKY or dispersed mutations. Those are mutations that are far away from each other. Experimentally, we set the threshold to be about 2K for the mutation. So if the mutations are the distance between two mutations in the genome is more than 2K, we consider them to be sparse. And then the mutations that are whose distance between them in the genome is less than 2K will be called clouds or also dense mutations. So we reason that if mutations are really far away in the genome, then the dependency is quite unlikely, while if they're close, then they're more likely to be dependent. So the idea now is to model the sparse mutations, the SKY mutations, simply as multinomial mixed model. Just model them as independent signature or processes. While for the mutations that are nearby, the mutation in clouds, we train a hidden marker model. Let me discuss briefly some of the biological findings that we obtained with Sigma. The first observation was that the characteristic of signatures often differ significantly in clouds versus sky. So, as an example, previous study considers the dependence of mutation density on the replication time. And when you look at this dependency in the by comparing the mutations that are in the sky, which are blue, and in the clouds in red, then for some of the signatures, those dependencies are very different. For signature 1, skies and clouds behave in the same way. But for signature 2, you can appreciate that early replication regions are enriched in the cloud signatures, while the late replication regions are enriched in the sky signatures. We can also learn something from the transition probabilities, especially looking at the transition between two different signatures. We can observe that signatures with related etiology are enriched from transition, such as signature 2 and 13 show often transition, homologous recombination related signature 3 and 8, etc. So this suggests that transition probability might give some intuition or at least hypothesis about the etiology of signatures for which are not well understood yet. Let me briefly mention a follow-up study. So this work is primarily driven by Roded Sharan's group, and it extended the model to a probabilistic model, and in addition to while looking at local dependency, it also takes into account the strand coordination, so looking at each strand separately. Let me briefly summarize this aspect of our studies. We showed that sequentially dependent models that model the sequential dependency, improve the assignment of mutation to predefined signatures. We also showed that clustered and dispersed mutation have different properties. And finally, we also showed that the transition between signatures can help to identify related signatures. This brings me to the next and very important aspect of the studies of mutational signature, which is that for many of those computationally derived signatures, we are still yet to establish what is the biological etiology of them. So in order to do so, we need to combine information maybe from both mutations and expression data. In order to explain the etiology of a yet to be understood signature, we developed two complementary methods. The first of the method, ECO-seq class, utilize gene expression models, while the second method is based on analysis of a mutated network. Our expression-based method is actually very simple, but we found it to be quite powerful. So basically, for the same set of patients, we assume that we decompose the mutational landscape of each patient into mutational signatures. So we know all the signatures and their strength. And for the same set of patients, we also know the expression of all the genes. So now we can take for any gene and for any signature, we can consider correlation of the mutation strength of signature strength and the expression. This allows us to build this genes by signature matrix where each entry corresponds to the correlation of a given signature with a given expression of a given gene. Most of the genes will not have really significant correlation with any of the signatures, so we filter them out, leaving only those genes which have a statistically significant correlation with at least one signatures. We then cluster those vectors, those signature vectors, and finally, for each coherent clusters of correlations, we analyze the genes that are in a given cluster through, say, Go enrichment analysis. So on the biological side, the thing that we really hope to understand and to get some insight about was the differences of the between the so-called clock-like signatures, signature 1 and signature 5. So those two signatures are known to be associated with patient age in many, but not all cancer types, and yet they're not correlated with each other. So if these two signatures are indeed clock-like signature, well, they will have to be ticking to different clocks. And I would like to understand what is the difference between these clocks. What are the two different time-related processes that are yet not necessarily all that synchronized between each other. Okay, so this next slide here shows the results of ECO-seq class analysis on breast cancer data. And in breast cancer, both clock-like signature 1 and 5 are present. And this analysis now allows us to have some insight into the differences. So one striking property, striking difference between those two signatures, signature 1 and signature 5, is that signature 1 is correlated with cell cycle and cell division. In contrast, signature 5 is actually correlated with nucleotide excision repair. So this is the relation of signature 1 with cell cycle is actually something that had been proposed before, since this signature is actually quite well understood, and it's basically from the basic biochemistry of the signature that I may not necessarily want to go into details right now. It is expected that the mutation that are related to the signature are likely to be accumulated with each cell cycle division. However, as mentioned, how we can explain how nucleotide excision repair may be related to time. And what we propose here is that the relation with time here is through environmental impact. That the environmental, mutation introduced by environment and most likely oxidative stress are being repaired by nucleotide excision repair. The elevated activity of this nerve pathway suggests elevated activity of those environmental factors, and one would indeed expect that environmental factors, the sum of the environmental factors, would generate signatures that accumulate with age. Encouraged by this analysis, by the result of this analysis, we then apply EcoSig class to the healthy control tissue of lung cancer patients. What we wanted to learn through this analysis is the impact of cigarette smoking on the healthy lung tissue. And we've been motivated to look at it because of a discussion that was in the literature about a potential positive or negative roles, depending on which paper you read, of smoking on the susceptibility to the COVID-19. So what we leverage here is that there is a specific signature related to smoking, signature 4. So for every patient, we can use this signature to estimate the amount of exposure to smoking the patient had. And then the exposure to smoking is really concerns the whole organism, not only cancer cells. So we can, although we measure the strength of the signature in cancer, we can understand how much exposure the person had, and we can look how that exposure to smoking influence the expression of genes in the control healthy tissue. And what we observe that the exposure to smoking is really causes the activation of cytokine-mediated signaling pathway. This is inflammatory pathways, and then subsequently does that inflammatory response pathway cascades further on to further level of immune response on the positive side, and on the reduction of ciliated cells in the lung on the negative side. So there's other interesting also conclusions that we draw from this analysis, and we hope to wrap up this paper for publication relatively soon. So far, we tried to understand mutational signatures by looking at them from the perspective of the associated changes in the gene expression. However, mutational signatures can be also related to mutation in specific DNA repair pathways, or generally be related to mutated pathways. So in order to explore this possibility, we developed or utilized our method, NetFix, which NetFix is a method allowing us to identify mutated network subnetworks that are associated with some phenotype of interest. This phenotype in our case would be strength of the mutation signatures. However, in initial application, we use as this phenotype of interest drug response. I will not have time to go into details of a NetFix algorithm, as not so simple like EcoSig class. However, on a very high level, I'll provide a very high level description. So we are given a set of patients, or for the drug response, set of cell lines. And for every patient or every cell lines, we measure the value of quantitative phenotype. For the mutational signature, that would be strength of mutational signature. For drug response, that was our first application, would be the response of a cell to a particular drug. In addition, For every individual or every cell, we know mutations within DNA of that cells, and also we can utilize the interaction network and the information about relationship between genes. So with that input, the goal of Netflix is to identify optimal subnetwork that is associated with a given phenotype, be that drug response, be that signatures. In case of drug response, it allows us to do further analysis, and we utilize it to look at the interaction between drug response. And if you are interested, I really hope you will look at our paper. Applying Netflix to mutational signature in breast cancer data, we have been able to identify mutated models associated with some, but definitely not all signatures. For example, signature 3, which is known to be associated with homologous recombination deficiency, has associated a model that contains BRCA1, mutation in BRCA2 and BRCA1, and TP53 for the cluster mutations, and BRCA1 and BRCA2 from the dispersed mutation, signature 3. So in this figure here, we order the patients according to the increased strength of the mutation as signature 3C, and the next row shows which patients have mutation in BRCA1, which patients have mutation in BRCA3, in TP53, and this is the summary of the mutations, showing the correlation between the mutations here and the signature strength. Other examples where we have been able to associate mutated subnetwork with the strength of specific signature include not TP53 subnetwork associated with signature 13. In the case of the dense or cloud signature 13, this network also contains the CHEK2 genes, and also associated of signature 2, the dispersed signature 2, with a subnetwork that contains PIK3A. A quick summary of this part. I showed two methods that are helpful to study etiology of mutational signature. Both of them, EcoSig class and Netflix, can provide helpful information about etiology. They are quite complementary. One is building on the changes in the gene expression that are associated with a given mutational signature, and the second, Netflix, is linking the mutational signature to perturb to mutated pathways. In addition, I showed that mutational signature can be used to study environmental impact on healthy cells or healthy tissues as well. Now, let me move to the last challenge I would like to discuss today. Namely, while defining mutational signature, we made an assumption that those signatures are additive. That for every patient, the mutation in the genome of that patient can be explained as a linear combination of mutations corresponding to each of the signature, each of the process. So this is, of course, non-negative matrix factorization is not the only way we can use to infer signature. However, this assumption of that signature are being additive is common to all the currently used methods. However, going back to the initial slide which I used to explain mutational processes in cancer, we can clearly see that those mutational processes are not necessarily additive. So, especially if we consider a combination of the what we call primary processes and secondary processes. The secondary processes, that is processes related to DNA repair, really act on the lesions that are produced by primary processes. So, for example, if if there was no way, if we had no lesion in the DNA, then it wouldn't really matter whether or not the DNA repair is functional or not. However, if, for example, we have like very strong impact of smoking and then also impact of DNA repair deficiency, this will create at the end a signature that is different than if we had a combination of UV light and then DNA repair deficiency. And if we go back and look at the database on the cosmic signature, we can really identify that there are 7 or 8, depending how you count, of mutational signatures that are being associated with mismatch repair deficiency. So a natural hypothesis is that this large number of mismatch repair associated signature might be exactly due to this composition of mismatch repair deficiency with some other processes, mutational processes that are happening. So first we focus on the proof of principle. How we can show that some of those signatures do indeed represent composite mutagenic processes. That is, processes that consist of one DNA damage repair process, where those DNA repair processes are different, DNA damage processes are different for different signature, and then common DNA repair deficiency that we assume would be common in number of signatures. How we can prove that this thing indeed happened? So our idea here was to define something that we call reprint as a way of representing mutational signatures. So a reprint is actually conditional probability distribution, where for each triple with left and right flanking residua, and under the assumption that mutation happened on the third position, on the middle position, we compute the conditional probability as what is the probability that mutation in this that this particular nucleotide is mutated in the remaining, in all possible ways, under the assumption that a mutation indeed happened. So this, notice that the reprint does not depend on 3 nucleotide frequencies in the genome, which is as opposed to the mutational signature. So basically, here's an example that if we have a triple ACA, what we are computing here is what is the probability that C is mutated to A, G, and T under the assumption that mutation in this triple happened. So if the what was different in different mutational signature was the primary process, the process that generated the lesions, and the DNA repair deficiency was common, then those processes should have similar reprint. And this is indeed what we see here as an example of 2 signatures that are known to be associated with mismatch repair. Those signatures, as you can see, are very different, but once we compute the reprints are similar. So we were interested to see whether there are other maybe signatures that have similar property. In order to do that, we consider all the 30 cosmic signatures and also additional signatures that have been determined experimentally, and computed reprints for all of those signatures, and then clustered those reprints. So what we can see here are 2 ways in which signature might be clustered, based on their signature similarity and based on reprint similarity. In colors here, I showed some of the signatures that are known to be related with respect to the secondary process. And as you can see, they are all distributed and not clustered together in the context of signature-based clustering, but in the reprint-based clustering, they nicely cluster together. So here's the cluster for mismatch repair signatures, and here's a cluster for homologous recombination deficiency. And here's one more cluster that we haven't deeply analyzed yet, but it appears that this is a cluster of signatures that are all somehow involved in repair, in damaging and therefore in repairing of bulky adducts. So reprint provided proof of principle that it is possible to identify those mutational signatures that correspond to composite DNA damage and repair process, where the the second step, the DNA repair process is the same. However, what remains still open, can we actually identify, can we actually provide describe the signature of those DNA repair, damage and repair processes separately? And this is actually the topic of our subsequent studies that will be presented on this upcoming recap, and I will only briefly summarize it here. So to summarize the basic idea, let's first assume that we have three mutagenic processes acting on cancer genome, and let us assume that DNA repair mechanism is intact, and it's reasonable to assume that all those three processes are independent from each other and that their effect is additive. In this case, we can simply apply non-negative matrix factorization to identify the corresponding signature. However, if DNA repair mechanism is deficient, then some of the lesions that caused by those mutagenic processes that normally would be repaired will not be repaired, and in which proportions are not being repaired will depend on a specific deficiency of the process. So in this case, in addition to modeling the mutagenic process itself, we also model the way in which some of the lesions are not being repaired, and that way is dependent on the characteristic of the repair deficiency. And we are excited to find that with this more complex model, we don't need 7 mismatch repair signatures anymore, but rather we've been able to uncover one mismatch repair signature, and we could compare the mismatch repair signature that is uncovered with our method with an experimental signature that was obtained by knocking out one of the components of the mismatch repair pathways, and those two were in excellent agreement. So let me, this brings me to conclusion of my talk. I hope I convinced you that study of mutational signatures are important for understanding of the role of mutagenic processes in cancer and healthy cells. I hope I've been able to convince you that there's a really great number of very interesting algorithmic questions that are related to this particular area. Some of them are related to what is the best way of defining mutational signatures. We use the study based on one of such definition, but that's not necessarily the only one. How we can identify the signatures that are present in a given cancer patient, meaning how we can identify the processes that are really contributed to the mutagenic mutation in a given cancer patient. How we can, given computationally derived signature, how can we think about on how we can link them to potential biological causes. There's a number of interesting questions related how to account for the context dependency of those mutations. The probability of mutations depend on the genomic region. And last but not the least, a very complex issue of the fact that mutagenic processes are not independent. And I was looking at some of the questions related of the dependency of mutagenic processes, but they also may depend on the activity of certain cellular pathways and other, other processes in the cell. So this is still a question that we just start to address, and many probably more technical and more algorithmic questions are likely to follow. Finally, the most important slide. I would like to acknowledge the members of my group who did the work that I described today, and also our collaborators. In particular, I would like particularly acknowledge Joachim, who did the work related to understanding of etiology of unknown signature, Damian Wójtowicz, who led the signature decomposition project together with Jan Choinka, and they were both helped by Rebecca Sartobazzo and Xiaoqing Huang. I also would like to acknowledge our collaborators, Max Leisesson, Rodert Sharan, and Fabio Vandin, and I would like to also especially mention the graduate student in Rodert Sharan group, Thais Seison. And I thank you all for the attention. I'll see if we have any questions.